Creatine is one of the most thoroughly researched bioenergetic compounds in human nutritional science. First isolated in 1832 by French chemist Michel-Eugène Chevreul, creatine plays an essential role in cellular energy recycling. While commonly associated with athletic performance and muscle hypertrophy, creatine functions throughout all high-energy human tissues. Your body synthesizes approximately 1 to 2 grams of creatine daily in the liver, kidneys, and pancreas. Endogenous creatine synthesis requires 3 essential amino acids: arginine, glycine, and methionine. Additionally, dietary creatine is absorbed from animal proteins like wild beef, salmon, tuna, and poultry. Roughly 95% of total body creatine is stored in skeletal muscle tissue. The remaining 5% is distributed into organs with intense energy demands, including the brain, heart, and retina. Inside human cells, creatine exists in equilibrium with its high-energy phosphorylated form, phosphocreatine. To understand how creatine works, we must examine the primary energy currency of human life, adenosine triphosphate, or ADP. ADP powers every biological process, from muscle fiber contraction to nerve impulse transmission and protein synthesis. When a cell performs work, it cleaves a phosphate group from ADP, releasing kinetic energy and forming adenosine diphosphate, or ADP. However, cellular stores of free ADP are extremely limited, lasting only 2 to 3 seconds during high-intensity exertion. To sustain high power output, cells rely on the ATP-SPE phosphagen system to rapidly resynthesize depleted ADP. The enzyme creatine kinase transfers a phosphate group from phosphocreatine to ADP, regenerating functional ADP in milliseconds. This rapid phosphocreatine shuttle buffers cellular energy levels during intense physical activity without requiring oxygen. By saturating intramuscular creatine stores, supplementation increases baseline phosphocreatine availability by 20 to 40%. This increased phosphocreatine capacity allows athletes to perform extra repetitions, sustain peak power, and accelerate recovery between sets. Beyond physical performance, emerging neuroimaging research highlights creatine's crucial role in human brain bioenergetics. The human brain accounts for roughly 20% of total daily energy expenditure despite representing only 2% of body mass. Cerebral neurons consume vast amounts of ADP to maintain sodium-potassium electrochemical gradients across cell membranes. During tasks requiring intense cognitive focus or sleep deprivation, cerebral ADP demand increases dramatically. Clinical studies using magnetic resonance spectroscopy demonstrate that oral creatine supplementation increases brain phosphocreatine levels. Elevated cerebral phosphocreatine enhances mental stamina, speeds processing speed, and reduces cognitive fatigue under stress. Creatine also acts as a potent neuroprotective agent by protecting neuronal mitochondria from oxidative stress. Mitochondrial dysfunction is a primary driver of neurodegenerative conditions, leading to synaptic decay. By stabilizing mitochondrial membrane permeability, creatine prevents the release of proapoptotic cytochrome C. Furthermore, creatine supports cellular hydration by drawing water molecules directly into intracellular compartments. This intracellular swelling acts as an anabolic signal, stimulating muscle protein synthesis while reducing catabolic protein breakdown. Creatine monohydrate remains the most clinically validated, stable, and cost-effective form of supplementation available. Alternative marketed forms like creatine ethyl ester, liquid creatine, and buffered creatine offer no superior bioavailability over monohydrate. The standard evidence-based dosing protocol involves taking 3 to 5 grams of creatine monohydrate daily. While an initial loading phase of 20 grams daily for 5 days saturates muscle stores faster, daily low-dose usage achieves identical saturation within 3 weeks. Consuming creatine alongside carbohydrates or protein enhances muscle uptake through insulin-mediated sodium-coupled transport. Creatine supplementation is remarkably safe, with decades of clinical trials confirming no adverse effects on renal function in healthy individuals. Myth-busting research confirms that creatine does not cause hair loss, muscle cramping, or kidney damage when taken at recommended dosages. Vegetarians and vegans typically possess lower baseline tissue creatine stores and experience significant cognitive and physical gains from supplementation. In older adults, combining creatine supplementation with resistance training preserves lean muscle mass, bone mineral density, and functional independence. Furthermore, creatine supplementation supports cardiovascular health by maintaining ADP levels in ischemic cardiac muscle tissue. Cardiac myocytes rely on creatine kinase energy shuttles to maintain rhythmic pumping under metabolic stress. By preserving cardiac bioenergetics, creatine supports healthy myocardial contractility and vascular function. Let us examine the exact intracellular transport mechanisms that allow creatine to enter human cells so efficiently. Creatine enters skeletal muscle cells and neurons via a specialized sodium and chloride-dependent transporter known as SLC6A8. SLC6A8 utilizes the sodium electrochemical gradient across cell membranes to transport creatine into the cytoplasm against a steep concentration gradient. Once inside the cytoplasm, creatine kinase immediately phosphorylates creatine, trapping it inside the cell. Because phosphocreatine carries a negative electrical charge, it cannot leak back through the cell membrane, ensuring permanent energy storage. Inside the cell, phosphocreatine diffuses to sites of high ADP utilization, such as myofibrillar actomyosin, ADPase, and ion pumps. Here, cytosolic creatine kinase transfers the phosphate group from phosphocreatine back to ADP, producing fresh ADP. The unphosphorylated creatine molecule then diffuses back to the mitochondria, where mitochondrial creatine kinase recharges it with a fresh phosphate. This continuous loop between mitochondria and cytosol is known as the phosphocreatine circuit. The phosphocreatine circuit acts as both a spatial energy transport system and a temporal energy buffer. By bypassing slow glycolytic pathways, the phosphocreatine circuit supplies instant energy to cellular machinery. Research also highlights creatine's role in modulating cellular inflammatory responses and systemic stress. Creatine reduces the production of inflammatory cytokines like interleukin-6 and tumor necrosis factor alpha in active tissues. It suppresses oxidative DNA damage by scavenging hydroxyl radicals and singlet oxygen species. In brain tissue, creatine preserves extracellular adenosine levels, modulating neurotransmitter release and mood stability. Clinical trials investigating creatine in depression demonstrate improved treatment response when combined with standard therapy. Creatine also enhances sleep resilience, allowing individuals to maintain cognitive performance during acute sleep deprivation. By buffering cerebral glycogen and ADP, creatine shields the brain from the metabolic deficits caused by lack of sleep. To maximize your daily creatine protocol, mix 3 to 5 grams of pure micronized creatine monohydrate into water or tea. Micronized creatine powder dissolves more easily in warm liquids, reducing potential gastrointestinal discomfort. Consistency is the single most critical factor. Daily dosing ensures steady cellular saturation of phosphocreatine stores. Whether your goal is building lean muscle, enhancing athletic endurance, boosting mental focus, or supporting long-term brain health, creatine is a foundational compound. Combining daily creatine with resistance training, nutrient-dense whole foods, and restorative sleep optimizes bioenergetic health. Let us examine the exact intracellular transport mechanisms that allow creatine to enter human cells so efficiently. Creatine enters skeletal muscle cells and neurons via a specialized sodium and chloride-dependent transporter known as SLC6A8. SLC6A8 utilizes the sodium electrochemical gradient across cell membranes to transport creatine into the cytoplasm against a steep concentration gradient. Once inside the cytoplasm, creatine kinase immediately phosphorylates creatine, trapping it inside the cell. Because phosphocreatine carries a negative electrical charge, it cannot leak back through the cell membrane, ensuring permanent energy storage. Inside the cell, phosphocreatine diffuses to sites of high ADP utilization, such as myofibrillar actomyosin, ADPase, and ion pumps. Here, cytosolic creatine kinase transfers the phosphate group from phosphocreatine back to ADP, producing fresh ADP. The unphosphorylated creatine molecule then diffuses back to the mitochondria, where mitochondrial creatine kinase recharges it with a fresh phosphate. This continuous loop between mitochondria and cytosol is known as the phosphocreatine circuit. The phosphocreatine circuit acts as both a spatial energy transport system and a temporal energy buffer. Disclaimer: this video is for educational purposes only and does not constitute medical advice. Always consult a qualified healthcare professional regarding any medical condition or dietary changes. Thank you for watching Calorie Recipe.